What I want to do is deal with one specific issue which was raised yesterday, and in talking with Maria, we realized that neither of us were covering it adequately in our presentations. And that is the issue of whether there is evidence of a potential for direct interaction with DNA. Because it seems to me, and Maria covered it quite satisfactorily in her talk, quite elegantly in her talk, and it is a statement in the ANSYS document, that the positive comet assay and micronucleus results that you see with TiO2 can easily be explained by oxidative stress. Induction of DNA strand breakage, induction of chromosomal, that will then lead to chromosomal breakage, which will lead to micronuclei. It seems to me that one of the key statements in the ANSYS document and how that will be viewed by, or would be viewed by IARC, will be viewed by ECHA, goes back to my earlier presentation of how genotoxicity data are used in carcinogen classification. I think that statement that we cannot exclude the possibility of a direct interaction with DNA needs to be looked at a little bit more carefully. So as Maria mentioned, and as came out in the ANSYS discussion, most of the time you can explain the most likely mechanism of action for genotoxicity to be due to oxidative damage resulting from an oxidative stress pathway. However, in several places, I've just clipped one of the quotes here. In several places in the CLH document, there is speculation that there may also be a direct DNA reactive component. So, for example, furthermore, a direct genotoxic mechanism with a direct interaction between DNA and TiO2 cannot be ruled out, in particular for nanoscale TiO2, since particles were observed in cells, including the nucleus. These comments seem to come from a number of publications. Not all of these were included. I think there's 4 of them that are given particular prominence, but all of these papers have some description within them that titanium dioxide particles were seen in the nucleus, or in 2 cases, as you'll see in a few minutes, were actually found associated with DNA. And you can see here there's a mixture of in vitro and in vivo studies. There's an inhalation exposure looking at lung tissue. There's a 90-day intragastric study looking at ovaries. There's an IV mutamouse study looking at liver tissue, and all the rest are in vitro. So how good is the evidence? Is there an alternative way to look at these data, this evidence? So in all of the above papers, some particles were seen to be associated with the nucleus. But often, and the authors do say this, often it was only an isolated cell. Now, therefore, the amount and the quality of the evidence on nuclear penetration is variable and inconsistent. In one case, a photomicrograph included in the paper, which was presented to illustrate nuclear invasion, shows a particle aggregated to approximately 1,000 nanometers. How can that possibly have penetrated the nucleus? Nuclear pores, we heard 39 nanometers mentioned yesterday. I found some data on the web which says it's 5 to 10 nanometers, depending on species. How does something of that size get into the nucleus? So the question is, did it get into the nucleus, or is it only being seen associated with the nucleus in the preparations that were put onto the transmission electron microscope? So the authors state that transfer of nanoparticles to the nucleus was seen, this is another quote from a paper, but was only observed in one cell. Or in another paper, particles were found in the cytoplasm of 3 different cell types, but were rarely found in the nucleus. So we've got really diverse descriptions. In every one of those papers, the in vitro and the in vivo, in every case, cells or tissues were fixed, embedded in epoxy resin, and ultra-thin sections observed by TEM. As Maria hinted at, and I will explain in more detail here, there are no quantitative dose-response data. In none of the papers do the authors tell you that they looked at how many nuclei contained particles at the low dose, the mid dose, the high dose, and did those frequencies of nuclei containing particles increase. There is no discussion of that. Why? Were they so rare that they couldn't be counted? Doesn't this raise a question about whether what they were seeing was a random chance event or whether it was biological cause and effect? This is about plausibility. This is Bradford Hill criteria. Are those observations plausible in terms of what is explained? More particularly, a group, Hondau et al. From Leeds, but it includes one of the world's experts on nanoparticle genotoxicity, Shireen Doak, who I spoke to about this issue. And she's worked with lots and lots of studies and reviewed lots and lots of papers on nanoparticle genotoxicity. And she says she has never seen any convincing evidence of a nanoparticle entering a nucleus under live biological conditions. What is happening and what Hondau et al. Raised as a serious concern is that when you section tissue for TEM analysis, particles in the cytoplasm can be moved by the microtome and accidentally, by chance, deposited in the nucleus or over the nucleus. These are called drag effects. And I want to show you this. I hope you can see it. This is from the Zhugan paper. Look on the left-hand side. I'm going to try to draw. This is the nuclear envelope, this curve. And this straight line down the middle is a drag mark. You can actually see it goes outside of the cell. However much you sharpen an ultramicrotome, it is never perfect. And when you're sectioning these epoxy-embedded preparations, you can get scratches. We know that nanoparticles, TiO2 nanoparticles, can get into the cytoplasm. There's no question of that. They are endocytosed, pinocytosed, whatever. And they sit there in lumps, and you're then going to section through them, and you're going to drag that microtome through the cytoplasm over the nucleus. Occasionally, you will drag a particle, and it will appear over the nucleus when you look down the TEM. That is probably why the data are so inconsistent, irreproducible, and there are no quantitative dose-response data. So is this evidence of nuclear penetration, or is it an artifact? I think we have a duty to consider the alternative interpretation. So it cannot be excluded that the photograph sections showing the presence of particle within the nucleus are actually views of the surface, are not actually views of the surface of the nucleus, i.e. that what you're looking at is a particle which is on the top of the nucleus rather than in it. One further point, there are 2 papers claiming that they isolated DNA from the livers of mice and from the livers of rats in these 2 papers. One was intra-abdominal injections, the other was 45 days intranasal. They claim that TiO2 was associated with the DNA extracted from those livers, or that titanium was associated, because that's basically what they were measuring. But in both papers, there is hardly any detail on how the DNA was extracted. Now, we know there are going to be particles within the liver. If you're not very careful about how you extract and purify the DNA, how do you know that you're not going to have some accidental, inadvertent contamination of the DNA as you pull it out of the liver from the TiO2 particles that are present in the cytoplasm in the tissue. There is not enough description to be able to rule that out as a possible explanation for why titanium was found associated with the DNA in those preparations. Do we not have a duty to consider that as a possible alternative explanation? So what I want to throw down in terms of a challenge is I believe that the data are not clear and not robust in terms of confirming that the TiO2 nanoparticles can actually penetrate the nucleus or interact with DNA. We find associations, but possible inadvertent contamination or drag effects cannot be excluded from the detail that is in the papers. But if that is taken to ECHA as evidence of a potential direct genotoxic effect, it is going to have serious consequences. So I think we need to consider these alternatives, and I thought it was worth taking a few minutes to try to get those points across.